Dr. Ain, you are muted, Doctor.
Sorry. Sekejap. Tak dengar lah pula. Muted. Hello? Hello?
Hi.
Can you hear me?
Yes, loud and
Yeah.
clear.
Yeah, okay. Mel? Okay.
Good, very good. Yeah.
Okay, so tadi tak dengar
Have to repeat back, Doctor.
Okay. A very good afternoon and welcome to INBIOSIS webinar lecture series 11 2021. I am Syarul Nataqain Baharum, will be your moderator for today's session. I hope the video has provided you with some background. So for those who are interested to collaborate with us, you are most welcome to do so. Feel free to browse our website and social media for the latest updates and info. I feel so grateful today that all of us are granted good health and time to attend our webinar session. There will be many more webinars to come, and I hope all of you will join again in the future. INBIOSIS webinar series is established to provide a knowledge and research experience sharing platform to ensure we are still connected with each other and stay motivated in our research despite these sudden pandemics. I'll now give some housekeeping rules for our webinar today. The lecture will takes about 45 minutes and the Q&A session will follow about 50 minutes at minimum. Probably will be longer depend on how many questions that we need to answer. Please submit the question in the Cisco webinar chat box and FB live page. I will read all the questions during the Q&A session. Before the seminar ends, I would like to invite all the participants to switch on the camera for a photography session. Please stay tuned until the end of the session to fill in the attendance form for e-certificate and Urus Bakat UKM credit. Without further ado, I would like to introduce our honorable speaker for today's session, Dr. RuAngelie Edrada-Ebel from the University of Strathclyde, United Kingdom. Allow me to share with you her CV. Okay, Dr. Edrada-Ebel received her PhD in pharmaceutical biology from Julius Maximilian Universität Würzburg. I hope I pronounced correctly, Germany. She did a postdoctoral fellowship at the Department of Chemistry, University of California, Santa Cruz, as a senior research fellow at the Institute of Pharmaceutical Biology and Biotechnology, Heinrich Heine Universität Düsseldorf, Germany. She currently a senior lecturer, associate professor at the Strathclyde Institute of Pharmacy, Biomedical Sciences, University of Strathclyde, UK, and head of the Natural Product Metabolomics Group at the Strathclyde Institute of Pharmacy and Biomedical Sciences. And currently, she's authored more than 140 publications, 4 patents on the marine natural products. She expertise comprise both natural product isolation and structure elucidation with the modern spectroscopy techniques. She has won many awards, including Planta Medica Most Innovative Papers in 2017 and International Fellowship with the Royal Melbourne Institute of Technology, and also the application of metabolomy on natural product research. Her laboratory is among the pioneers in establishing the application of metabolomics for targeting analysis of bioactive secondary metabolites. At present, she has a series of ongoing projects on the application of metabolomics to identify and biotechnologically optimize the production of bioactive secondary metabolites in marine-derived microorganisms. So without further ado, the floor is yours, Dr. RuAn. Thank you.
Thank you very much, Dr. Syarul. Can you hear me? Can you see me?
Yes.
All right. Yes. So thank you very much, Dr. Syarul, for the introduction. And thank you very much as well for the invitation and giving us opportunity to share our research to you this afternoon in Malaysia. My share button at the moment is gray.
Can you share this? Yeah, can you share the slide?
No, my share button is gray.
Oh, okay.
I cannot click on my share button.
Oh, okay. Wait.
I think now I'm presenter. Okay, good.
Okay.
I've got
Okay.
it.
Okay. So we still waiting for the slides. Okay.
Right. Can you see
So
it now?
yeah. Now can see.
All
Okay.
right.
Okay. Good.
Okay. So
Okay.
this afternoon's webinar, it will be about the use of analytical data in a metabolomic-guided approach, particularly in targeting the isolation of bioactive natural products. So what do we mean by concatenated analytical data? This is practically the fusion of data sets that includes, for example, a combination of UV and IR, NMR and mass spec, or NMR and UV, MS and UV. So there are a lot of combinations wherein you could fuse your data sets. But for this afternoon webinar, I will be introducing fusing data sets from mass spectrometry and NMR. So just a short introduction about our group. We are part of a multidisciplinary consortium. So we work with a lot of European countries. So we do our sampling together with a group in Greece and Iceland. Natural product chemistry is basically done in our laboratory. Then we've got genetic and metabolic engineering that is basically done in Norway with the University of Norway. And then bioactivity testing is also done in Strathclyde, but we've got collaborations with SMEs like Pharmac in Norway and Axum in Italy. Then we've got genomics that is done in Germany, University of Würzburg, and then industrial bioprocessing. This is in collaboration with SMEs in Scotland, as well as in Iceland. So we don't only collaborate within Europe, but we also have our global partners. And for today's webinar, I will be presenting work with Brazil, that is the Sao Paulo State University, Iceland with Mattis, and the third example that I will be presenting will be a work together with Malaysia. So why metabolomics? So we use metabolomics as a tool for drug discovery. So in the literature, it's usually coined as natural product metabolomics or secondary metabolomics. And as you would know, when we use metabolomics, macros are embedded with natural product databases, and that will enable us rapid replication studies. Part of metabolomics approach also involves multivariate analysis softwares to investigate patterns in data. And when we say patterns in data, our main objectives are the following. So we use it as a selection tool to find the best plant or microbial strain to work with, because we would like to determine chemical novelty. Or we could also use it as a selection to optimize our process conditions. So which means we could integrate metabolomics data with process variable data sets. And we could, for example, link chemistry with fermentation, because we do work with a lot of microbial fermentation. This includes fungi and bacteria. Most commonly used in our group is to investigate bioactivity, which means we could compare chemical profiles of bioactive and inactive extracts. And this is quite practical, because then we could actually target the bioactive metabolites in the first isolation work. And finally, together with molecular biologists, we could then integrate the genomic data with the chemical data, and this would confirm the expression of biosynthetic gene clusters. And this is very important, for example, when we'd like to investigate biosynthetic pathways. So metabolomic tools, we believe, will provide a clear and well signposted map to the target destination. So as we know, natural product research could be comparable to walking through a maze system. So With metabolomics, we believe that this could be better than a road and traffic signs. So in natural products research, we don't want to be ending in a roundabout within roundabouts. Or we want our standard operating procedure to be just going in one direction and not made up of several arrows directing us to various paths. So I do know, we are aware that when you're doing natural product research, finding novel compounds needs a little bit of luck. Yeah. But I think with metabolomics, we could target where we're going to. So for example, again, this is a confusing sign here, keep right, but it's actually going left. Yeah. So when you're doing natural product research, we just need to be aware of road surprises, but definitely we don't want to be going the wrong way and going back. We don't want to be depleting our resources. We want to work them, we want to use them efficiently and not wasting them. So I think this is the main role of using a metabolomics approach. So this is our development workflow. I think this is also being used by a lot of laboratories. So we start our isolation and culturing when we're working with microorganisms, and then we do extraction. So this is the same way you do with plants as well. And then the extract are subjected to metabolomic analysis. And as I've said in the beginning, we are using both mass spectrometry and NMR. Parallel to this, we do taxonomic identification and genome mining, and we also do bioassay screening. So all these steps are done at the same time or in parallel. So I mentioned about the analytical platform we're using. So we're using both mass spectrometry and NMR spectroscopy. And the reason behind that, both analytical platforms have their advantages and disadvantages. So when we're working with NMR spectroscopy, we do need a larger or higher concentration of compounds to be able to detect these different complex metabolites in a sample. However, the great advantage is that the sample preparation is quite generic. So for example, we just use a universal solvent like dimethyl sulfoxide, which will both dissolve polar and nonpolar components. Another great advantage of NMR spectroscopy is it's very reproducible. It really doesn't matter when and where you measure your samples. As long as the samples are stable, you do get the same spectrum. However, mass spectrometry, the great advantage is you could detect low concentration compounds. You could even go down to picogram levels as long as the compounds are ionizable. However, disadvantage is it's not as reproducible as NMR, which means it's highly dependent on the method you're using, calibration, ionization capability of the compounds. And there are also specific methods to prepare the samples because most, for example, you're working with electrospray ionization. This is only limited with polar components, and you could only, of course, dissolve your compounds in polar solvents. So you cannot use, for example, chloroform or dichloromethane for your non-polar samples in this case. So this is an example of a predefined metabolomics workflow. We always start with mass spectrometry, although we're doing a fused data analysis. So because it's always an untargeted type of analysis, we always have both positive and negative switch mode. Then the mass spectral data is processed. We use MZ-Mine2, but there are also other processing softwares available in the market or in the web. The main purpose of the processing, we should be able to do peaking, the convolution, the isotoping, alignment, and most important, formula prediction. And this is a prerequisite when it comes to the replication. So I mentioned in the beginning that we should be able to embed our macros into a database. So in this case, we use, for example, anti-marine, or we could also use Dictionary of Natural Product. There are also some available databases on the web, like GNPS, but our main concern is how vast these databases are, okay? And then once we've got the replication and the data is being processed, then everything is put into multivariate analysis. At this stage, we start to fuse our data. So here in multivariate analysis, our NMR data will come in, as well as our bioassay data. So in this case, we could use SIMCA, which is commercially available. Metabolomics is free, is a freeware, and everyone could access it from the web. Or each group could also generate their own R packages. Okay, so using R packages, they could also generate their own programs. Okay, and the main purpose of doing multivariate analysis is to find trends and visualize your data in a more logical manner. Once we've got our results here, we are able to target our compounds. They could be novel. And if they're novel, then we do again a structure search to make sure and validate that we've got the novel compounds. And finally, we get a full structure elucidation of our compounds. So for today's webinar, I will present to you 3 application projects. So first is optimizing the production of fish antiparasitics from Algoriphagus marincula. So this is actually a bacteria isolated from the geothermal plants in Iceland. And then accessing the seasonality or Prisonema intermedia from the Brazilian Cerrado flora. And finally, comparing Malaysia and New Zealand honey. So my third application project is going back to Malaysia. All right, so the first sample project, so this is in collaboration with Mattis in Iceland. So this was mainly done by Dr. Bella Sanchez. So she's a PhD student from Portugal. So just a brief background on the project. So the antiparasitic activity was observed from 7 days cultures of the Algoriphagus marincula, and they were inoculated by single colony, but affords very low biomass. So that was quite a problem for us regarding scaling up the bacteria. So when we did the scale up in our laboratory, we lost the antiparasitic activity from the 7 days culture because we changed our inoculation method. So this time we use a 10% inoculum in spite of a high biomass yield. So we need to investigate what is really happening. Our hypothesis is that using our fused datasets, we will be able to validate the production of the bioactive compounds, which we think is occurring at an earlier growth phase when we're using the 10% inoculum technique. So our main objective is to improve sample clustering and group classification when looking at data sets to be able to prove our main hypothesis that the antiparasitic compounds are being produced at an earlier stage when using the 10% inoculum. So our main question is, can we validate the optimization method during the scale-up on the production of these antiparasitic isobranch alpha? So these were the type of compounds that Bella was able to isolate, and they were active against fish parasites. So these are actually salmon parasites. Okay. So if we directly fuse the data from 2 analytical methods, are we able to confirm on which is the optimum day of the production of these type of compounds? The second aim is to increase the yield of these antiparasitic compounds. So we don't want to use the single colony method. Yeah, definitely if we use that, then our yield will be very low. So we still want to maintain the 10% inoculum. Okay, so just a brief description of how we fuse our data set. So MS and NMR data were scaled using this equation. So we use the peak area of the NMR, which are in chemical shifts in ppm, as block 1. And the second block is the mass overcharge peaks from our mass spectrometry data. So we put these two blocks together, okay, and we get the sum of the standard deviation of the respective blocks. And this could then be managed statistically using multivariate analysis. If you would like to have a more detailed description of how we do this, okay, you could find this in our publication. So it's an open access in scientific reports. So this is a study on the secondary metabolomic profile on Tithonia diversifolia. Okay, so let's go back to the project of Bella. So here, what I'm trying to present here is the data before and after fusion. So here, and the scores plot for A. So this is mass spectrometry data on its own. We do see a trend from day 1 to day 7, but obviously we could see that there is a difference in chemistry between the 10% inoculum, which is the blue spot, and the single colony, okay? So we do only observe a common chemistry in the first to the third day of inoculation. The same thing is observed in the NMR. So day 1 to day 7, they were quite separated. But here we could see more specifically that at day 3, okay, there is a merging of the data set, which means there is a kind of similarity in the chemistry when it comes to day 3. When we fuse the data from day 1 to day 7, okay, we could see here again that day 3, there is a similarity in the chemistry. However, it was still not clear to us what is happening because we could see a big difference between the 10% inoculum and the single colony. So we did a log transformation of our data set, okay? And we could see here that day 3, 5, and 7, okay, although is still separated from the day 7 from the single colony, we could see here that our antiparasitic compounds are starting to be produced in day 3, but then on day 7, the yield of these compounds are going down. And that is actually the reason why we are losing the bioactivity on day 7, because our compounds are already produced on day 3. So that is quite positive, and our industrial partner was very happy on that because rather than waiting for 7 days for the production of our compounds, we could then use day 3 to start harvesting our compounds at a relatively better yield than a single colony procedure. So regarding replication, we could also hear, for example, from mass spectrometry, this is actually the problem that's happening here, that the replicates were not really reproducible. So in mass spectrometry, you can see that the replicates are far from each other. And NMR, they were quite close. They're still coming in the same cluster, okay? But they are not as 100% together, okay? So they are not 100% similar. And these errors could have something to do with extraction errors, okay, as well as fermentation errors. So using a fused data set, these errors are being corrected. So not only do they appear in the same clusters, but they are also coming together. So the error of extraction and fermentation are corrected during concatenation. So that is the great advantage of using a fused data sets. These errors are also being inhibited in this case. So let's go to the second project. So this is using as well MS and NMR. to access the seasonality of Prisonema intermedia from Brazilian Cerrado flora. So this plant is used by the locals to treat to treat as an So the plant infusion is found to possess antiseptic, antimicrobial, antihemorrhagic, and anti-inflammatory properties, and it is widely used by the local. They usually use the barks and the leaf infusion. So inspection of the MS and NMR spectral data between the samples collected at various seasons do not indicate any clear differences in terms of biomarker components. So the main objective here is to find out where are these anti-inflammatory compounds being produced, not only where, but when. Yeah. So we do have an hypothesis that the major compounds are accumulated in a certain season and is highly dependent on the major environmental factors. So the objective is to pinpoint the seasonal biomarkers from a unique or more unique perceptible constituents, particularly in the production of the anti-inflammatory triterpenes. So again, to compare the data sets, so here again, we've got mass spectrometry. So the green is the summer season, okay? And we've got here the red as the winter. So we've got the separation of the type of metabolites being produced in the winter and the summer season. However, here, you can see that spring and autumn are all together. When we get into the NMR, the summer season and the springs are separated, and we have an overlapping of the metabolites produced during the summer and the autumn. So separation is only observed during spring and winter. In the fused data set, the production of the metabolites become more obvious. Okay, so here we have a separation of the summer. We also have here the winter and a very relatively good separation between the spring and the autumn. Okay, but okay, so the main conclusion is that we got a clear separation of the summer samples, while we've got a sloping trend from winter to spring. To find out what are the biomarkers, so we look at the summer, which was always good, where we always get a good separation. Okay, so here we've got the PCA and the OPL-SDA. And we could also find that according to their molecular weights, we could see our triterpenes being produced in the summer. And this is the NMR data as well. Okay, we could also see the production, okay, of the aliphatic triterpenes in the summer. Now, what Anna did was to look at as well the concentration of the relative abundance of these triterpenes that is being produced in the summer. And she concluded that there is a higher pentacyclic triterpene content with lupain and oleunin structures in the summer, as seen here. Okay, although this triterpene structure is commonly produced both in the spring and in the summer. Okay, so only these type of triterpenes are being produced in the summer. However, during the spring, what is a very good biomarker is the higher concentration of triglycosylated flavonoids and galloflavonoids. So here are the example of compounds that were found as biomarkers during the spring. So the take-home message here, as you would see in the data when we look at them without fusion, is that the spring metabolites were always overlapping with other seasons. But using a fused data, we could actually define which types of metabolites are being produced during the spring. And finally, to conclude Anna's result, okay, regarding the production of the metabolites in summer, we could then make a conclusion that triterpenes are being produced at higher temperatures with a solar radiation between 2,000 to 2,500 units. And lower humidity is also very important, with a rainfall between 0.15 to 0.10 millimeters. So these are now the optimum conditions that would produce the anti-inflammatory triterpenes. And these served actually as a very good conclusion for Anna's work in answering her objectives. So finally, let us compare Malaysian and New Zealand honey. So this was done by Dr. Yusnani. Okay, so just a brief background. So the Malaysian honey exhibited bioactivity against breast cancer cell line, while the New Zealand honey was found inactive. So what is actually the difference between the two honeys? Inspection of the MS and NMR spectral data, however, did not indicate any apparent difference in terms of the major components. So if you look at the data, we would say that the major components between New Zealand honey and Malaysian honey are the same. But what is actually making the Malaysian honey very active while the New Zealand honey inactive? So again, using a few data sets, will these be able to improve the clustering and correct the differences between replicates to define the bioactive biomarkers? So our main objective is to pinpoint the low-yielding bioactive biomarkers from the more perceptible constituents. So just imagine, we know honey is rich in sugar. So that is actually what we're seeing in both samples from New Zealand and Malaysia. So we are thinking that we've got low yielding bioactive biomarkers. So they could be present at microgram or nanogram levels that is giving a very good potency against breast cancer. So before fusion, this is the data set. So we could see, for example, a very good separation between Malaysian honey and New Zealand honey. Okay. So that's the NMR, the same thing. So even a better separation between HRMS data. So these are all OPLS data. So the main problem here, we have certain samples that are actually almost crossing the border. And I think this is the reason why by inspection, we are seeing the same thing between NMR data in Malaysian and New Zealand honey. We can see here, okay, so this is the loadings plot. So it tells us the different features in the NMR. So for example, here on the Malaysian honey, we could see a high concentration or density of spots between 3 to 6 ppm. So we know, but 5 to 6 ppm, we have the phenolic compounds. And as you would see here, you could almost see that the same spots are also observed, except that you have here the occurrence of aliphatic functional groups in New Zealand honey. So this is the type of problems that are occurring in our NMR data. Okay, the data sets actually, although we see this separation on the scores plot, okay, the data set really doesn't define a big difference between the Malaysian and the New Zealand honey. In the mass spectrum data, we do get a separation, the scores plot, but if you look at the loadings plot, we almost see the same type of compounds, okay, between the Malaysian and the New Zealand honey. So here we have molecular weights between 150 to 420. Maybe these molecular weights are smaller from the New Zealand honey, which is between 150 to 285. But this, from our opinion, is not really conclusive. So after fusion, we got some surprises here. So here we've got the Malaysian honey. It's well separated, but we have again a separation between the New Zealand samples. So this one, I think, is from the New Zealand honey from the south, and we have here the New Zealand honey from the north. However, it became more conclusive that the New Zealand honey has higher lipid content as represented by the higher density of darker blue dots. So you can see here that there is a higher density of darker blue dots, while the higher density of lighter green spots, as presented here, indicated lower molecular weight compounds. In the Malaysian honey, we could see a higher sugar and polyphenolic content as represented by the higher density of lighter blue dots, while the higher density of yellow-green dots to orange spots indicated higher molecular weight. So we could see a more conclusive difference between the Malaysian and New Zealand honey when we fuse our data sets. Now, quite interesting is the separation, the natural separation between the New Zealand honey, between the northern and the southern region. Okay, so here, for example, in the northern islands, so NZ2, so you have a higher density of the blue spots. Okay, however, and these are represented by higher concentration of the acetylated compounds. However, in the New Zealand honey coming from the South Islands, okay, we have the higher concentrations of the light blue spots, and they are representing the higher phenolic content. So we would say that the South Island samples were more comparable to the Malaysian honey. And maybe that is the reason for some of the crossover when inspecting the features in the NMR data between New Zealand and Malaysian honey. So to summarize and conclude, fused data validated the optimized production and harvest method for the target metabolites, as we saw this in the first example in the project with the Iceland bacteria or antiparasitic compounds from Algophriphagus marincola. The infused data also improved the linearity of the production and the presence of the metabolites against the respective parameters considered in the study. And finally, the fused data also resulted to better clustering and corrected the differences between the replicates to define the biomarkers as exemplified by the 3 projects. So finally, I would like to thank my group, as well as the collaboration with the various universities, as well as with the SMEs involved in this project, particularly Pharmac, and of course, our funding agencies. And finally, I would like to thank you for your kind attention. And yes, we're ready for questions. Hello?
Okay. 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 Thank you, Ruan, for the very enlightening presentation on your 3 different projects. So basically, the projects are using very high, approach, analytical approach, which is you have the HRMS in your place to work on to work with, and also on the NMR for the validation of the results. So we open for question. If there is any question in the chat box, I'll read the questions. Let me check. Okay, just to start the ball rollings, I have some question for you, Ruan.
Yes.
Okay, so as we know that on the honey production, because you're talking about Malaysian honey as compared
Yes.
to the New Zealand honey, so it's like very interesting to know the details. So we know that there is a variable factors that influence the honey productions, the compounds in the honey as well. So in Malaysia, I think the environmental factors very influence the production, right? So
Yeah.
in Malaysia, we have more maybe raining season as compared to the New Zealand. So and then also the flowers, different type of flowers as compared to New Zealand. How that's why we have, you can see the difference um, compound distribute in both honey. So as you mentioned that in the Malaysian honey, they produce more on the high molecular weight compounds as compared to the New Zealand one. So can you elaborate more on that? What type of compounds that produce in Malaysia honey as compared to the New Zealand?
Yes, so when we look at, use, we're able to isolate also some of the compounds, and the type of compounds she was isolating were either flavonoids with multiple sugars, but while in the New Zealand honey, where she also did some isolation work, she was isolating more triterpenes in her work. So that was a big difference. Although, yeah, so the multiple glycosylation of the flavonoids were causing the high molecular weights of these compounds. So she would see, for example, that the phenolic compounds will be glycosylated at 5 positions. So that will give her a molecular weight between 500 to 800. While the triterpenes, normally that were coming from New Zealand, okay, they will be present as free triterpenes, so they will be unglycosylated. So they're still considered to be medium molecular weight compounds. So, but she will be getting molecular weight compounds of around 300 to 400. So not as high as um, those isolated from Malaysia, which will go up to 800. The other problem with the New Zealand honey, where she had difficulty working out, was that there was always this high lipid content. So when during her isolation work, she would always form this emulsion, and it was hard to. We know it should be also rich in sugar, but I think the sugary content would emulsify with the lipid content of that is found in the New Zealand honey, which didn't occur when she was processing the Malaysian honey. Mm-hmm.
Even the physical appearance also different, right?
Yeah, they're different. But
Ah,
when you look at the NMR, you could only see the sugar content, and there were no big difference. Yeah. But in multivariate analysis, if the data were not fused, you could see that um, the differences that the NMR was defining were there. Okay, but it was not conclusive because, as I showed you, there were only blue uh, spots, which is representing the aliphatic components of both. Yeah, so yeah, we cannot conclude anything when the data were not fused. But when we start infusing the data, then it become more obvious what were the difference between the Malaysian and the New Zealand honey. And she was also able to define the biomarkers for the anti-cancer activity, which was obviously the phenolic compounds. Mm-hmm.
have you test on that biological activities for that, the anti-cancer compounds in
Yes,
both
she
honey?
did that. Yes, in
So
both honey. Mm-hmm.
so which one is much more better, I mean, in terms of the results, higher production?
Yeah, it's the Malaysian honey that's
Ah,
easy.
that
Yeah, so the New
yeah.
Zealand honey were inactive. Mm-hmm.
So
So that's
we are glad to hear that.
yes,
So we
yeah,
should eat more Malaysian honey.
right, yeah. I think it has also something to do with the type of course, the type of plants, the geography. Yeah.
Yeah,
So
yeah.
yeah, use also look at the type of plants surrounding the territories where the honey was um, collected. And that is what actually made the honey unique. So, of course, the plants you find in Malaysia, you will not find in New Zealand.
Yeah, true.
And vice versa.
Yeah, true.
Right.
Thank you for that explanation. Okay, I go to the chat box. Okay, there have a question here. Great presentation by Dr. Ruan. Did you perform metabolite identification in the honey study using MS?
Yes, yeah. So we could only perform the replication using MS, not NMR. Yeah, because in MS you could put that in a database, and there's no database available now. Well, there are some coming up databases for NMR for certain compounds, but it's not as intensive as mass spectrometry. So regarding the replication study, we're still using mass spectrometry, not NMR. So NMR just helps us with the replication to validate the replication result in the mass spectrometry. So for example, the replication result is telling us you've got quercetin. So we go back to our NMR data and see, could we see the signals that represent quercetin? Yeah. Or are we getting to see other set of signals? So that is how we do the replication between mass spectrometry and NMR. So initially we use mass spectrometry, NMR we use to validate what we see in the MS data.
Yeah. Thank you for the answer. Okay, the next question by Dr. Imelda. In your experience, which would be more efficient for the profiling, MS or NMR?
As I've mentioned, so for initial profiling and the replication, MS will be better. Okay. NMR, the problem with NMR is that you won't be able to identify compounds down at microgram or nanogram levels. And NMR always offers a more complex data set. So you have your overlapping. Okay, so you don't really get a good separation. Unlike in mass spectrometry, if you do an LCMS, then the compounds are relatively separated. Yeah, so I would say, yeah, mass spectrometry is still a better technique for chemical profiling.
Okay, the next question by Dr. Hamidun. Can you apply the same technology to bacteria?
Yes, yeah. So my first example was a bacteria. So Algaerophagus marincola is actually a bacteria. And as I've mentioned in that example, we were having difficulty in finding out why our 10% inoculum was not working while the single colony was positive in the bioassay. And then using the concatenated data, we saw that the target metabolites were already produced in the 3rd days. Yeah, which were not relatively obvious when the data were not fused together. Yeah.
Okay. The next question by Dr. Hamiza. Thank you very much for the excellent talk. Regarding the difference between Malaysian and New Zealand honey. Okay, another question about the honey. May I know what are possible factors that might lead to the differences? Is it due to the environmental factors or different honeybee species between both countries?
I would say it's more in the environmental factors, not really the bee species, because the bee will just forage on whatever is around their environment. Yeah. So we know, for example, Malaysia is a humid tropical country. New Zealand, although northern and southern New Zealand are already very different. So southern New Zealand, yeah, this is a big question in AU's project, is that southern New Zealand is more, it's very near the cold Antarctic region. Okay. And then, but however, it's still producing phenolic compounds comparable to those in Malaysia. And then we've got the northern island, which is actually more comparable to Malaysia because that's the more tropical region. Yeah, but they are still producing a different type of honey than Malaysia. It's just because the plants available for the bees are different. Yeah. So the bees will just, no matter what type of bees they are, okay, they will just forage on whatever is around their environment. Okay, and these plants are actually um, affected by their environment as well. So I would say it's the environment, not the type of bees.
Okay, thank you for the answer. The next question, may I know why Simca was chosen over other chemometric techniques?
Okay, why Simca? I think Simca is a very good software because of the loadings plot. So it has that feature or to be able to pinpoint, okay, what metabolite would be considered as a biomarker. When it comes to, well, we just compared it with Metabolomics because my students also started working on Metabolomics. So Metabolomics is very good in terms of visualization, particularly on the scores plot and separating the different samples. However, it really doesn't define the target, what is making the separation. Okay, so for example, my presentation on Anna's results. So that is a combination of using both Metabolomics and SIBCA. Yeah, so all her scores and loadings plot were done using a SIBCA. But then the box plots, looking at the relative abundance of each of those metabolites, were done from Metabolomics. So I think the best way really is to use the best in both um, methods. Yeah. Well,
Is
the
it
only disadvantage. Oh, sorry.
it's okay, it's okay.
Go.
Continue.
Yes,
Sorry, yes, yeah.
sorry.
You can,
The
yeah,
only,
you can continue, sorry.
okay, the only disadvantage of Simca is that we pay for it,
Yeah,
and Metabo Analyst is it's a freeware, so everyone could play around it.
that's
Yes.
actually my comment. If you have money, you can use Simca. But if you don't have, you want to have a budget on other things, you can use the Metabo analyst. Yes.
Right, yeah. So the nice thing about Metabo Analyst is they have these graphics as well. They separate wherein it tells you directly which are the discriminating biomarkers. And then if you click on this discriminating biomarkers, it's not a loading plot. It's just a graphics. It's like going to the VIPs, the very important parameters. Okay, so if you click on that, okay, it will tell you which are the discriminating features, and then it will generate this box plots. So that one is also quite easy to use and quite practical. Yeah.
Yeah, true, true. Okay, the next question. Thank you for your great presentation. May I know how you concatenated the data between MS and NMR? How you relate all the different platform?
Okay. Um, yes, right. So you can do that by Excel. Okay, so just like I have shown on my slide. And then, so you have your block 1 and block 2, and then you get the standard deviation of each of the rows, okay? And then you get the sum of the standard deviation of both blocks, right? And these standard deviation values could be moved into any multivariate analysis program. However, if you're using Simca we could automatically fuse the data sets for you. So just put block 1 and block 2, and it will do everything for you. So you don't need to do it manually by Excel, wherein you have to process each row individually in Excel. But if you have Simca, yes, only that Dr. Syarul said is that you need to pay for it. Is that it does everything for you automatically. So it will fuse the data sets for you. So you just have to define which one is your block 1 and which one is your block 2. Yeah.
Thank you for the
And yes,
answer. Yeah.
right. And for more detailed uh, methodology on fusing data sets, yeah, just go to our scientific reports paper. Yeah.
Okay, thank you for the answer. Okay, the next question by Hakim. Razak, which gave you the insight to use fused data and concatenated approach to answer your questions?
Okay, so we want the diffuse data and concatenated approach when our data sets in mass spectrometry and NMR are not conclusive, or the conclusion we're getting from both data sets are against each other. Okay. So if, for example, in the NMR it's saying, so you could see that obviously in Anna's project. So in the mass spectrometry there, for example, is a good separation between spring and summer. But in the NMR, there's a good separation between summer and winter. Okay. So it's not conclusive when certain biomarkers are really being produced. And we want to know, for example, well, in Anna's project, she was only interested on the anti-inflammatory triterpenes. However, for example, if she wants to look at the phenolics, which is also very good in antioxidant activities or anti-cancer activities, then she needs to define which season are they being produced best. And that was not really conclusive when the spring metabolites are overlapping with other seasons. So which is really the best one? Could we get the harvest of these phenolic compounds? For the triterpenes, it was quite obvious because the summer were always separated from the rest of the other seasons. Okay, but to define the different biomarkers, that only gave us the answer with diffuse data sets. So the trends could always be better on the diffuse data sets. It also depends on the type of questions you're answering. And as another thing is that, for example, in the project of use on the honey, as well as the project of Bella on the microorganisms, we could see a separation of our samples when we don't fuse the data. And that has something to do with the errors that are difficult to correct, like extraction errors, fermentation errors, which is automatically, well, I say automatically corrected in when you fuse the data sets. Mm-hmm.
Okay, thank you for the answer. The next question, second question by Akil. Did you identify all the metabolites in each platform before you connect it?
No, we didn't. So there are some unknowns. Yeah, we don't know if these are new compounds. Yeah, but we were not able to identify all of them. So for honey, would say we have identified around 80% of the compounds, so 20% were not identified. Mm-hmm. Yeah, but at least from the replication data from the NMR, we were able to find out what type of compounds they were. So whether they were phenolics, aliphatic, lipid-like, or terpenoids. Yeah. So that's actually the advantage of using NMR. Get to see the chemical groups. Yeah.
Okay, the next question by Dr. Tan. How do you perform metabolite annotation using MS?
How do I perform? Again, sorry, I didn't understand the question.
Metabolite annotation using MS.
Metabolite annotation using MS. Is the question something to do with molecular formula prediction, or?
I'm not sure. That's the only question here. Metabolite annotation using MS.
What do you mean by metabolite annotation?
Maybe Dr. Tan, you can ask the questions. Maybe you can unmute your mic. Dr. Tan, are you here?
Hi, here, Dr. I. Can you hear me?
Yeah.
Dr.
Yes.
Great
Yes.
presentation. Yeah, actually, it's how do you actually perform metabolite identification using the mass? Yes.
Okay, identification. Okay. All right. So metabolite identification using mass spec. You could use the okay. Of course, we use the formula prediction. Okay, so the number of oxygen, nitrogen, and double bond equivalence. So that's how we do the pre-identification. But if you want a very good identification, just using mass spectrometry without NMR, of course, we need to do MSMS. Okay, so a very good procedure, which of course I didn't introduce in this talk, is that, for example, using molecular networking. So that will also give you an idea how many of the derivatives are actually related using MSMS dataset. Okay, so that's another type of experiment. So MSMS will give you actually a full identification, well, relative identification of your compounds. Yeah. Yeah, but at this stage, what we do in the preliminary screen, when we want to use, because it's just the replication study, is we match our NMR with the mass spectrometry data. Okay. And when we already know our targets, okay, predefine the targets, then we go for MSMS analysis, and then we can do molecular networking as well and do an identification. But we don't really do a full identification of our compounds just using mass spectrometry.
Okay, I hope that answer your question, Dr. Tan. Okay, the next question by Nur Hilwani. Thank you for the enlightening talk. I'm interested to know the type of Malaysia honey used in the study. Was it tualang honey? Is there any publication from your group that you can suggest for further reading? Thank you.
Yes, it's tualang honey. That's true. No, this is still the publication has just been submitted, so hopefully we get it accepted.
Yeah. Please share with us once you, please share with us once you get it accepted.
Yes, well, we'll share it. Yes.
Okay, the next question from the FB Live by Dr. Goh. Is the Malaysian honey from the stingless bee? Why choose Malaysia and New Zealand honey?
No, it's not from the stingless bee.
Okay.
Why choose Malaysian? Yeah. I really don't know why. Sorry.
Okay. The next question, why choose Malaysian and New Zealand honey?
All right. Okay. So I really don't have any idea why New Zealand honey. Well, it was obvious that it was Malaysian honey because my student came from Malaysia. New Zealand honey, I don't know how she established her contact with New Zealand. Yeah, but she had a company. She contacted New Zealand and they were able to deliver honey. Maybe the reason why, this is just mainly my speculation, is that in Malaysia, maybe New Zealand honey is very commonly used as well. Is that?
Not really.
No?
Not really. Yeah,
Okay.
It's much more expensive.
It's more expensive, or maybe that's the reason why she wants to compare Malaysian honey versus New Zealand honey, to prove that, come on, Malaysian honey is cheaper and it's more effective.
Yeah, I think so. Yeah, that's the real answer, I think.
Yeah.
Okay, another question by Dr. Tan. How many metabolites that you managed to annotate using MS alone in the honey study? How confident is your annotation? I think more to identification. I think.
Yeah, more identification. Yeah. Oh, a lot. I don't know how many. So I think if you look at the just the mass spectrometry, I think because it will also annotate or identify metabolites at picogram level. I think it's almost more than 5,000.
Wow.
Yeah. How confident we are? We could only be confident with those that we could see on the NMR. Yes, those are the high intensity peaks, those that could ionize better. So for example, you have 5,000 annotations. Not all these 5,000 annotations will give you good intensity peaks. Yeah, so maybe the first 100 or the first 50 peaks with good intensity or a good peak shapes could be proven as good identification. But beyond that, beyond the hundreds or beyond the 70, yeah, I won't say that they are that we would be confident on their annotations. The p-values when doing this processing, this mass spectrometry processing, it would also give you the p-values, and you would see that the p-values, okay, will increase So which means it will decrease the confidence level as you go down the 5,000s. So you get very good p-values in the first 10 identifications. So your p-value will be less than 0.01. Yeah, but then as you increase the as you decrease the intensity of this peak, the p-values will also increase. Okay, so perfect identification, I would say p-values less than 0.01. Yeah, so that will give you a 99% confidence, and that will be the first 50. Okay, or even so p-value of 0.05 will be the first 50, and the p-value less than 0.01 will be the first 10 or 20 peaks that is being identified. Okay, so that's the confidence level in terms of statistical significance.
Yeah, thank you. And then there's a lot of work need to be done to identify the compounds, right?
Exactly. Right. Mm-hmm.
Okay. The next question by from the FB Live by Dr. Aizat. Thank you for the insightful talk. Any specific honey types bees from either Malaysian and New Zealand? What type of sugars that can be identified from these samples? Recently, a scientific report paper suggests the presence of trehalose sugar in Malaysian, Brazil and Australian honey, which is low GI beneficial for controlling the sugar level in diabetic patients.
Yes, that's correct. So yeah, I could only say yes to that. Yes, it's, yeah, we did find a lot of these sugars, but then we were really not interested on these sugars. So because we were looking at the sugars when so it was used as a blank by used to do her bioactivity anti-cancer activity test, and of course obviously they were inactive, the trehalose sugar against cancer, but of course it has, yeah, as you said, they have low GI benefits for controlling sugar levels in diabetic patients. That could be true, but yeah, we don't have any studies on that. Yeah.
Okay. There is any trehalose present in your sample?
Yes, I think we saw that, but as I've said, we didn't go on to further studies in it. So yeah, because we actually in the first step of our extraction, use already try to discard all the sugars. We just want the organic components and find out, okay, where's the bioactivity in these organic samples. So the aqueous samples were obviously inactive. Yeah, it doesn't have anything but just sugars. Okay, and this trehalose sugars are one of them, but yes, we were not interested in them anyway, so we didn't look further into those. Mm-hmm. Regarding the type of honey, type of bees, is that the question?
Yes, is a type of the honey types and
Yes.
also the bees type, Malaysian and New Zealand.
Yeah, we didn't know what type of bees we yeah, that was worked on or from where this honey was collected. We only know that they were not stingless bees.
Normally for the tualang honey, normally is from the apis species.
Okay, right. Yeah.
Okay, I think that's all the questions in the chat box. Okay, I think we almost finished our session for today. Okay, it's just some announcement to be made. Okay, we have come to the end of the webinar. So I would like to thank Dr. RuAngelie for the very interesting subject, and I'm sure has opened up our view and give an idea on how you can bring up your research to the next level. Today webinar has recorded about in our WebEx about 104 and in the Facebook Live about 34 participants. I also would like to thank all the participants for spending your time with us today. Please take please stay safe and take good care of yourself. But please, before we end the talk, please switch on your camera so we can have photo session. Hi, Dr. Shukri.
Hi, Shukri. How are you? Nice to see you.
So please switch on your camera so we can have the photo session with our speaker.
Hi, Winnie.
Yeah, Winnie. How are you?
Okay, sebelum tanya ya, ada Aisyah sudah habis menjawab ujian kedua.
Yes.
Aisyah boleh ke
Secretary, are you ready for the photo session? Okay. Okay.
Everyone ready?
Okay. So please smile.
Please smile. 1, 2, 3.
Okay.
One more. Wait, yeah. Okay, one more. 1, 2, 3. Okay, thank you.
Okay. Okay,
Thank you, everyone.
Okay, thank you, everyone. Dr. Ruan, please stay. So we have another session with you after this.
All right.
Okay, thank you. So thank you, everyone. Bye.